Eukaryotes like us are typically made up of many different types of cells. However, all of us started from a single cell. We had a sperm that fertilizes an egg that becomes our first cell. And that cell divides into clones, and all of the cells in your body have the exact same genetic identity. You'll have the same genome in all of your cells. However, we have different types of cells. You have stomach cells, heart muscle cells, liver cells, and they all function differently. And the way that they function depends on the proteins that they express. And so we actually have to turn genes on and off in our different types of cells to have different functioning cells. Eukaryotes have a lot of different ways to do this. So we can do this by modifying the DNA itself, making it either too tight to transcribe or loosening it up so that it can be turned on. We can do this through transcription, choosing which genes get transcribed and which ones do not. If a gene can't be transcribed, it can't be expressed. RNA processing, as well as methods of post-transcriptional things like translation or messenger RNA degradation, can determine even if transcription occurs, which genes actually get translated into proteins. So we have many steps in our process from DNA to RNA to protein where we can change something and either turn a gene on, meaning that it's going to get expressed, or turn a gene off so that it does not make a protein. So let's start at the top with chromatin. So chromatin is our DNA and our histone proteins. And there's a couple of different changes we can make to chromatin that will either make it more likely to be transcribed, such as histone acetylation. So histone is the proteins that the DNA gets wrapped around in the nucleosome. When you add acetyl groups to the histone tails, those are these little green dots here, it actually loosens up the DNA. So this is a very compact heterochromatin, and it's getting loosened over here to a euchromatin, which can then be transcribed. So this is going to kind of turn a gene on. Another thing is called DNA methylation, and this is the addition of methyl groups to certain bases in the DNA. What this does is it typically is going to turn a gene off, so it's going to go the other way. It's going to take some loose DNA and make it into tight, more dense DNA. When the DNA is wrapped really compact, it's not really accessible for transcription. The transcription factors and the RNA polymerase, they can't get in there and do transcription. So this is going to eventually turn a gene off. An example of DNA methylation in humans is actually X inactivation. People with two X chromosomes typically turn one off. It gets inactivated. And this is because that X chromosome gets methylated to the point where it's not going to be used for transcribing any genes. This is typically called epigenetics. So epigenetics is the transmission of traits by mechanisms not involving the DNA nucleotide sequence. So what we just did, adding methyl groups or acetyl groups, those didn't change the DNA, the A's, the G's, the T's, and the C's. All it did was make it either tight or loose, and it changes which genes get expressed. So in this mouse, you can see these two mice are identical. So they are exact clones. However, the maternal diet, which was different for these two mice, resulted in different expressions of the fur color genes. And so one mouse gets the normal kind of brown color, and then this other mouse turned out orangish gold because the epigenetics, the acetylation or the methylation of the DNA, was different for these two individuals, even though their gene sequences are the same. So epigenetics is one field of study that we've been looking at in the last couple of decades, which might be able to explain situations where you have identical twins, and one of the twins is treated for schizophrenia, which has a genetic basis. However, the other one does not have schizophrenia. And that might be because the gene that causes the schizophrenia was methylated differently in those two individuals, even though their genetic sequence is the same. So the next step in DNA to RNA is transcription. So we can see there's a lot of regulation during transcription. So here is our DNA transcript. So you can see that this is the promoter. This is the transcript here that's going to get copied. And there's the poly A sequence, which is going to tell the RNA polymerase it's time to stop. Along with that, there are many control elements, and these control elements are part of the DNA. You can find proximal control elements. So this direction is considered upstream of the gene. So proximal control elements are going to be very close to the promoter. And then you can see a break here. Enhancers are going to be important. These are distal. Distal means far away. And so these are upstream of the actual gene, but they're going to tell the gene when it's supposed to be turned on and when it's supposed to be turned off. All genes are going to need transcription factors in order for that protein to get made. So RNA polymerase cannot start at a promoter without the assistance of transcription factors. So what we're going to do is we're going to see how these control elements activate transcription factors. And that's how we know when to turn a gene on and when to turn a gene off. So the proximal control elements would be really close over here. But what we're going to focus on right here is the enhancer. The enhancer is a distal control element, and it can be very far away from the gene. It may not be very close. It can be actually very far away. In order for a gene to be turned on for the transcription to begin, the enhancers need to be activated by an activator. And what we're going to see in these images is they're going to use some color coding. So you can see that these enhancers are kind of this orangish brown, and the correct activators are also this orangish brown. So this is one way that different cells can have different genes turn on because they have different activators. All of the cells in your body have the same enhancers because they all share the same DNA. But the cytoplasm and the nucleus are going to have different activators depending on which genes we want to turn on or turn off. So the activators will bind to the enhancers. That's going to tell this, okay, it's time to turn the gene on. Then we'll see a bending protein, because again, this can be very far away, that's going to actually bend the activators to become close to a group of mediator proteins. And then those mediator proteins are going to interact with my transcription factors and my RNA polymerase. And then this is going to go ahead and start initiation of transcription to go ahead and turn this gene on and transcribe it into messenger RNA. So let's look at an example. And here we've got two different types of cells. So inside of a cell, we have a gene for albumin. Albumin is made in the liver, and it is not expressed in the eye. Inside of both of these cells, we also have a gene for crystallin. Crystallin is going to be a protein found in the lens of the eye, but it doesn't need to be made in the liver. So you can see how the function of two different types of cells is going to depend on the proteins they make. Both of these cells have both of these genes because we all started as one single cell that just got cloned. So in the liver, we see that the albumin gene is expressed. Here are the enhancers for the albumin gene. We've got yellow, gray, red. And the available activators are yellow, gray, and red. So those are going to combine to the correct enhancers, mediator proteins, and then eventually transcription factors to transcribe the albumin gene. We do not see brown or pink. So there's a limited number of enhancers, but by using different combinations of enhancers associated with different genes, we can make sure that the liver is turning on the albumin gene, but not the crystallin gene. So this one is on, and this one is off in the liver. In the eye, so we have a lens cell in your eye, the available activators are different. We have pink, gray, and brown. The gray one does actually bind here, but because we don't have yellow and we don't have red, then the albumin gene is off. It does not get turned on. However, because we have pink, gray, and brown to match the enhancers on the crystalline gene, this one gets turned on. And so we can see that even though these liver cells and the eye cells have the same genes, they have different proteins called activators, which bind to the DNA enhancers and turn on and off different potential genes. We can also see things happen after transcription. So even after we make messenger RNA, we still have to process it. So there is a processing called alternative splicing, in which the same piece of messenger RNA can produce different protons, different proteins. So here we've got a troponin gene. So this is going to code for pieces of muscle cells called troponin. And you can see that there are 5 exons, 1, 2, 3, 4, and 5. And then these intervening pieces here are the introns that are not going to get coded for. Well, there are 2 different ways to splice this troponin gene into 2 different forms of troponin. And it just depends on whether they're going to choose 3 or 4. So we see regulatory proteins that are going to come in at this step, and they're going to make the choice about which introns and which exons get used. So in this option number 1 over here, the regulatory proteins chose 1, 2, 3, and 5. So this will be a protein with regions 1, 2, 3, and 5. Over here we have option 2, which chose 1, 2, 4, and 5. And so because these have slightly different pieces of messenger RNA, they are going to have slightly different structures of the protein and slightly different functions. So we started with 1 gene and we ended up with 2 proteins. The last step of protein synthesis is translation. So even though we made an RNA, and even though we got the RNA into the cytoplasm, it still has to get translated into a protein to be expressed. So there are some methods in which we block the initiation of translation. So this is going to prevent the binding of the ribosome, and that's going to prevent this gene from getting expressed. Sometimes we are doing this for a single gene. Sometimes we initiate translation for many genes to be stimulated. So for example, when you first become a fertilized egg, the initiation for translation factors are all simultaneously turned on to start the process of gene expression and getting translation and transcription going. The lifespan of the messenger RNA also matters. The longer the messenger RNA is available to ribosomes, the more of those proteins are going to be expressed. We have methods of using enzymes to degrade the poly A tail, remove our 5' cap, and then you can see these little Pac-Man here are going to eat these messenger RNAs and degrade it. So these are called exonucleases because they're breaking it down. Lifespan of messenger RNA inside of the cell is usually not very long. Specifically, bacterial or viral messenger RNA is going to be removed very quickly. So those proteins tend to not get expressed because they get broken down very quickly. Whereas if we can make a messenger RNA look like it belongs there, then it won't be broken down as quickly and it can make more of its proteins. Most of the human genome doesn't actually code for proteins. So what we figured out is that all of the genes that we have in our DNA, only about 1.5% of our DNA actually codes for proteins that express functions. In the past, scientists have called the rest of our 98.5% of our DNA as junk DNA. However, we know that's not true. We've got control elements and we've got something that controls, and we have some fractions of our DNA that can be transcribed into something called a non-coding RNA. So these are not messenger or transfer or ribosomal RNAs. These are just really small types of RNAs that have different functions in the cell. And just because they don't code for a protein doesn't mean they're junk. So here are a few examples. We have microRNAs, which are miRNAs. And what these will do is the miRNA will bind to a complementary piece of messenger RNA, and it will either physically block a ribosome or break that RNA into small pieces. And both of those are going to prevent translation, so this is going to typically turn a gene off by preventing it from being expressed. Similar to that, we have small interfering RNAs, or siRNAs. These are similar in size and function, but they're slightly different in the way that they're developed. And these are going to block gene expression through RNA interference. And we actually use these in laboratories as means of disabling genes to study their function in knockout studies. Another one is the Peewee interacting RNAs, or piRNAs. These are going to work at the chromatin level by forming heterochromatin. So this is going to turn off gene expression for these genes by preventing those genes from being transcribed in the first place.